Hello, I'm your host, Ray Dogum, and welcome to VibeCast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel in important drug development. VibeBio's mission is to find every cure for every community. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guests today are co-founders of Likarda, Lisa Stehno Bittel and Dr. Karthik Ramachandran. Welcome, guys. How are you today?
Good.
Awesome.
Doing well. Thanks for having us.
Yeah, thanks for being on the show. Really appreciate it. And today we'll talk about Likarda, the company that you guys co-founded, why you did so, and some of the advancements in cell therapy and whatnot.
Great.
So why don't we get started with some introductions, and maybe Lisa, you can go first and just tell us who you are and what brought you to this point in life so far, really quick.
Yeah. Thanks for having us. Yes, I was in academia for years, and prior to that, I was actually a physical therapist. So my career has kind of taken a tortuous path, but always when I found an opportunity for something I enjoyed doing, I tended to jump on that opportunity. So I was in the university setting for over 20 years. Karthik was one of my PhD students, the first bioengineering PhD student that I had. And we had some patents together. Eli Lilly showed some interest in those patents. And so that's when we spun the company out in 2012, initially, mainly as a virtual company out of my research lab. But then we quickly transitioned after Karthik graduated into incubator space in the university. And now we've grown into our own space.
That's awesome. Thank you for that. And Karthik?
Yeah, so my background's actually originally in biology, and when I came to University of Kansas going into biomedical engineering, I got excited by the research that was happening in Lisa's lab around diabetes, and so convinced her to take on her first engineering student, and really got into kind of drug discovery, drug development, but focused on cells, and had a passion for also taking products from concept to maybe market. And so also participated in some business courses and a fellowship kind of around the business side. And when we decided to spin out Likarda, I thought, well, this is a perfect opportunity. I was already a broke grad student. I'd be a perfect broke entrepreneur to start something new. And we thought together it would be fun to see if we could take our project and kind of move it to market.
Yeah, and we all know, well, I mean, people who do this know how difficult it is to get something from an idea to, in the market. So appreciate you, trying to do that, working on it. So what motivated you, Lisa, to actually, go ahead and start Likarda and say, let's do this company? And then let's talk about what Likarda is actually doing after that. Yeah.
Okay, sure. Karthik and I, as I mentioned, were working in diabetes and really trying to find a better way to conduct islet transplants for people with type 1 diabetes.
And what are islet transplants?
Okay, great question. Yes. So islets are the insulin-producing cells of the pancreas, and pancreatic transplants for people with type 1 diabetes don't work. It's an autoimmune disease. The autoimmune aspect just destroys the transplanted pancreas. But at that time, there was some success around just taking out the insulin-producing cells and transplanting those. It was hit or miss, and there was a real shortage of donor tissue for that. In addition, the person had to be on lifelong immunosuppressors, and those come with their own set of problems. So we were working on better ways to improve that process. Karthik's dissertation, as part of his dissertation, he developed a microplate that allowed us to make miniaturized islets so that they could live off diffusion better and function better in the recipient. We were at the American Diabetes Association meeting presenting this, and Eli Lilly saw what we were doing, they're scientists, and asked if they could use those plates to do drug screening for new diabetes drugs. So we got a grant from them. That's what you do in academia. You get grants. It went well. We published a paper with them, and then they wanted to start sending a few blinded compounds for us to actually test for them. So I was very excited. I sent a proposal, a scope of work up to my superiors in the Research Institute at the University of Kansas, is where we were located. And I immediately got a phone call from the vice provost saying, Lisa, you can't do this. First of all, my expenses at that time, I didn't pay rent, right? I'm in a university setting. I didn't pay utilities. My graduate students were at that time making about $24,000 a year. If I just went with my costs, it was going to be a really cheap project for Lilly, not anywhere near market rates. So I had padded the budget a bit, put in some profit. And they said, we're a state institution, we're a nonprofit, you can't have a profit line in there. And there are rules in the state of Kansas that says that you cannot compete with any Kansas company. And there still are CROs in Kansas, contract research organizations, companies that do contract research. It wasn't a direct competition with them, but yeah, there might have been a little overlap. So at that point, the vice provost said, if you want to do this, you've got to spin it out as a business. The university had an incubator with full business personnel there. They did market analysis for us. They set up kind of the business plan, looked at budgeting. And so we spun out, as I said, initially kind of virtually from the lab, but then moved into incubator space at the university. So your question is, what motivated us? It was really, I think we were incubating a business within my research lab for a while without even knowing it. And then we already had a client right there. So why wouldn't you do it? And within 18 months, we were revenue positive because we had these clients lined up. And we ended up not really working in diabetes the way we thought we would. The real strength came in cancer. So making miniature tumors for drug screening became a big business for us very quickly, and we were able to grow it for the first 5 years of the company.
Wow, that's amazing. It's cool that it was a sort of a natural progression too. It wasn't an idea you just thought of and wanted to build a company. You actually had a process already in place to start collecting revenue essentially from Eli Lilly. That's amazing. So in addition to that, so if you think about what techniques can improve the efficiency of cell and biological therapy technology, so is what you're doing improving the techniques for therapy, or is it just to diagnose or identify certain compounds that could?
So that's the next phase. We're taking all of the profits that we made as a CRO and still putting them into cell therapies, specifically diabetes, because that's always been our passion, is to cure a disease rather than treat the symptoms, right? And so about 2016, we started out as the only company that could do high throughput drug screening in 3D spheroids or organoids. And so we were able to charge a premium. And as I said, we had a nice client base. Then the large CROs started moving into the space with their own technology and even moving beyond the plate assays that we had developed. And so we were losing business. We were getting undercut in pricing. Had to make a decision. And this was our first pivot. We've made 2 pivots with the company. This was our first pivot. Are we going to put a ton of money into the CRO and build out a full CRO, which is a service, built company, and those are hard to scale, expensive to scale? Or were we at that point going to take our first investor? So and really focus on the cell therapies. So we were 5 years into the company before we took our first investor. And that was the decision we made. Again, always staying with our mission. Our mission and our passion was always the cell therapies. The CRO was a means to the end. So we, at that 2017, kind of slowly let go of the CRO work and allowed our scientists to focus solely on cell therapies. So what came out of that then was what did the cell therapies needed? And as I mentioned, patients had to be on lifelong immunosuppressors. So we started working on delivery systems for cells that protected the cells from immune attack so that they did not have to be on immunosuppressors. And that's really the beef of Likarda. The gem of Likarda now are many different delivery systems for cells and biologics.
Have you since sort of let go of the CRO services operations completely, or are there still some operations? Yeah.
So it's different now, and Karthik can speak to this. We still provide a service to clients, but it's now building a customized hydrogel delivery system for them.
Very interesting.
And so it's more of a partnering now rather than just providing research data and handing it over to them.
Yeah. So one of the unique things that happened when we kind of pivoted into that cell therapy space, we recognized we were frustrated by the delivery systems that were out there. Then there's really only one delivery system. It's all based on alginate, which is seaweed. Use alginate in food gastronomy to be able to make things like vegetarian caviar. It's kind of like Jello, effectively, but you wouldn't really want that in the body. It's foreign to the body. It doesn't work well. Over the years, people tried chemistry to make that work. It just wasn't working. And as scientists in the lab, we decided, well, there are better technologies out there that still could offer this, but maybe it's not yet at a point where it can be an encapsulant. So these things look like little caviar beads when you make them. But one of the things we found are materials that are used in things like wound healing, in cosmetic fillers, in joint supplements, things like hyaluronic acid. We all know that. We hear this from L'Oreal. They sell hyaluronic acid. These things are safe when you inject them into the body. They're actually naturally occurring in the body. But there was no way to take that before from a hyaluronic acid paste into these injectable microbeads. And that's really the innovation we brought at Likarda. Was we invented a platform for how do you encapsulate cells and use these kind of next generation materials rather than making a modification on a current material. Let's just move to better materials. That solved a lot of our own internal problems when we were thinking cell therapies. But as we continued to work with these hydrogels, everything from hyaluronic acid to PEG to polyvinyl alcohols to a number of other hydrogels, we found, wow, this is starting to become a platform. And as we were publishing this data, we started to get some phone calls and emails from other folks who said, okay, this is really interesting. Similar to Eli Lilly, this is interesting. We've got a cell therapy. Could this be a fit for us? And that's kind of when we started to think, okay, now our platform has an opportunity to partner with others. To grow a therapeutics company would mean needing to raise $100-200 million to go through phase 1 or phase 2 clinical trials. But to partner with a company who has that kind of funding, already developing the cell therapies, but recognizes they need this innovation, that's what really then kind of bolstered us into this business of bringing delivery systems to cell therapies.
So interesting. So for the audience, would you mind kind of explaining hydrogels and their advantages for cell therapy delivery or other types of drug delivery?
Yeah. So a hydrogel is basically Jello. So Jello is made of gelatin. It's 98 or 99% water. It's mostly water. And when you mix the hydrogel, or let's say gelatin with water, you let it set, it becomes kind of a hard solid. That's what a hydrogel is. So you could do that with things like hyaluronic acid. We actually have chemistries where you can cross-link those, so that way they become more permanent cross-links. So this is more on the chemistry side. The benefits for cells is when you do put this kind of Jello-like coating around a cell, it protects it from the immune system. So when you have a cell that is foreign to the body, so a transplant, it's coming from a different donor, it's coming from a different cell source, it's going into the body, you want to make sure that the immune system doesn't attack it. Typically, you're taking an immunosuppressor for that. If you think about heart transplants, kidney transplants, most of those patients are taking immunosuppressors, so that way their body doesn't kill off the organ. That's what you would normally have to do. But in this case, with the hydrogel protecting, the immune system can't actually come in and attack the cells. So they just kind of hit that surface and bounce off. The benefit of the materials is when the immune system sees those materials, they also see them as more, natural, more normal to the body. So they also don't attack that material like they would something that's also a foreign material. And so what that allows is protection from the immune system, but because it is mostly water, you're still able to get nutrients in. They just kind of diffuse in, and you're able to get all the waste and products that the cells produce. So in the case of diabetes, something like insulin, that can come out of the microbeads. One of the benefits then of working with these beads, again, I always say they kind of look like caviar when you see them. They are injectable. So now you go from needing major surgeries, major medical device procedures to something that can go through a needle or a catheter.
That's really interesting. So you're building these like advanced sort of jello protection mechanisms to deliver these cells. And what's cool, what you said is you can still have nutrients permeate through the hydrogel and allow the cell to survive the journey into the right location. And what types of, you mentioned diabetes as one indication disease, but I think, this can also treat all types of diseases. Do you want to kind of talk about your folks there?
Yeah. So we originally started in diabetes because that's what came out of Lisa's lab. We are looking at materials that can protect, that don't fall apart. Diabetes is a long-term disease. You want the materials to stay around as long as possible. One of the interesting things we found is a few of the materials we worked with while we were testing them were falling apart. If you were only focused on diabetes, you would say, well, that's actually failing. We don't want it to fall apart. Then the immune system can come and attack them. But what we found in our research was the body isn't attacking these things when they're falling apart. The hydrogel is completely still safe. And we started to then explore, well, could that be useful in something like osteoarthritis to deliver stem cells into the knee? And that's really kind of what took us onto this other trajectory of, looking at osteoarthritis. We've been in the liver space. We've looked at the cardiac space, CNS. Even one of the popular spaces we're in now is looking at delivery of cancer-killing cells, so things like CAR T technologies to solid tumors. So we are working in a number of spaces now.
That's interesting.
And I'll just add a little bit about how we differentiate from some of the other hydrogel companies that are out there. As Karthik mentioned, alginate, which is a permanent, because it's foreign to the body, there's nothing that breaks it down. So it lasts a long time. So it can be used as an immunoprotectant, but it can't be used as a delivery system. And we took these chemicals that are already being used in medical devices or dermal fillers. They're already being put in the body. They don't cause a reaction, a negative reaction. And the body has enzymes or uses hydrolysis and can break them down so that they can be a delivery system. And what we found after that was that not only can they deliver cells, and as Karthik mentioned, cancer is a good example. The immune therapies that are cell-based against a tumor, those cells have to bind to the cancer cells. So you can't just keep them in a ball and have them secrete things. They actually need to get out of that encapsulant. So that's where we have a big advantage that nobody else has. And what we've happened in the last year is that people are seeing that, well, this can also deliver more than just cells. We're having trouble getting antibodies to the target site to stay there long enough to have an effect. We're encapsulating antibodies now. We can encapsulate gene therapies in their vectors so that they can, if they're placed at a site, these beads are very sticky. So once you put them someplace, they stay there. And then we can design them. They can slowly release whatever the active ingredient is for 3 weeks, for 6 weeks, for 3 months. The client tells us. Yeah,
what's like the
client tells
longest
us
time frame?
how long they want them to stay there. And then we'll design the delivery system to match their needs.
Is there like a physical limit on how long it can do, like a 20-year drug or something?
Probably not.
Okay.
No, they will break down over time. We've gone out 18 months and still the beads were intact and functioning. The cells inside were functioning. Those are animal studies. We're going to be doing a dog diabetes study. We're working on getting our manufacturing in place for the FDA. But we'll be doing a diabetic dog study in family pets. And at that point, we'll be able to see what is the duration of our strongest cross-linked hydrogel.
Awesome. I appreciate that. Let's jump back to 2017 when you were raising funds with investors for the first time. How were your conversations like with them? And I'm sure they were quite happy that you were already revenue positive.
Yeah, it's a funny story. And again, a kind of an unusual story for people starting companies. I was out pitching Likarda, again, trying to find new clients for the CRO. The CRO was struggling, but that was our revenue source. And in Kansas, there's a man named Peter Worth, who was a founder of Chemworth, which is one of the largest generic active ingredient manufacturers in the world, and very well connected, obviously, in the pharmaceutical industry. And I knew that he was going to be at a Kansas university on a trip. And so I asked the president, I called the president of that university and said, could I get 30 minutes with him? They said, sure. So they set it all up. And I started doing the pitch, and he said, well, we had some diabetic dog studies already, some data, good data. He was like, well, why don't I just give you money? I'll just invest in that. And I said, no, that's not why I'm here. No, I need more, contracts. I need more revenue coming in. Can you help there? Well, but Lisa, if I just invested in this, then you wouldn't have to do the CRO and you could focus on this diabetes. And he's so excited about our work in diabetes. And I just kept saying no. And in retrospect, I think there was some arrogance on my part. I was very proud of saying we had not had any investors at that time. So after that meeting, I came back and, again, the CRO still every day trying to bid out projects and getting underbid and just frustrated with things. And Karthik and I talked about it. And I said, maybe we ought to think about this a little bit more before we just say no. So we went back to Peter, and he is our only investor. It's Worth Family Investment Association, and he's been absolutely amazing. I could not have asked for a better partner, a more supportive partner. He makes introductions for us where he can. He's our best cheerleader, and we are now just starting a Series A fund. So we will be bringing in some new investors for the first time, but so much of this journey, I didn't talk about how we launched. As a CRO, you've got to have a lot of expensive equipment. We needed robotic liquid handling machines. That came from my family. And so I think about how many great ideas are still left on the lab bench because, one, they didn't have a family with the resources to be able to launch them. It was a significant amount of my folks' retirement that went into this. And then they don't have a Peter Worth. And, those were just being in the right place at the right time. There's some luck associated with this. And I never want to take that for granted, that we've worked hard, but we also just had some amazing things happen to us that helped us along the way, and amazing people that helped us along the way.
Wow, that's a great story. Yeah, it sounds like Peter definitely changed your mind about taking in investor money.
Yeah.
And, it's interesting that a lot of, co-founders or founders, they start with their own money or their own family's money. And that's a big decision to make. It's a huge sacrifice and a huge risk. And many people just say, no, forget it, not worth it. They join big pharma and that's the end of that. But there's so many good ideas like this.
That. Yeah. It was like jumping off a cliff without a parachute. I didn't sleep at night, a full night. For years, I didn't sleep a full night's sleep until we at least until we were revenue positive. And then I kind of could calm down a little bit. But it was such a risk for my whole family. And that's, I think, another point to make is that when you start a business, it's not just you. Your whole family is starting that business. Specifically your immediate cluster, your partner, your children. Everybody is putting a lot on the risk and on risk. And again, another lucky thing was for me, my husband had a great job. We, as the incubator people, were working through, is this going to work? And what's the market look like? And doing all that work before we actually launched, Doug and I practiced and just lived off his salary because we knew there were going to be times where I wasn't going to make any money, and realized that, yeah, we can do this. It's okay. But again, just the example of how lucky we were, but also it is a sacrifice by more than just you.
Yeah. And I'm glad it's, starting to pay off. And now you have, the Series A round that you're raising. So good luck on that. And
Thank you.
I'm sure people who are listening might be interested as well and just learning more about that. So I encourage them to check out the description, show notes to find you, Lisa, or, reach
Absolutely.
out to VibeCast, me. Happy to connect you guys. What is Likarda's next critical value inflection point in the company, would you say?
Well, right now we are working on this Series A, of course. And what the Series A will do for us is really build out that funnel of clients coming in. As I mentioned, we're now expanding into biologics, antibodies, peptide delivery, gene delivery. And we even have a client now looking at, they want a really extended release of a small molecule, which we didn't think that would be of interest to anyone, but they want like a 3 to 4 month release of a small molecule in a specific location. So a lot of that 15 million is to expand our own portfolio of hydrogel technologies. We've got about 50 different formulations that we have created and manufactured up to date. But to expand that, another advantage that we bring is cryoprotection. So cells especially, it's a real problem in cold chain storage and shipping, and the hydrogel will help protect the cells. And for biologics, there's a problem that they tend to precipitate or they aggregate and they're no longer functional, a peptide, for example, and the hydrogel can keep that from happening and again, help with storage and shipping. So we need to build out that. But a real inflection point is we have one licensing opportunity in animal health is to build out more licensing. So once we get a client through a project, a feasibility project, and we get a hydrogel that works for them, a formulation that works for them, then it really is licensing down the line. So adding to our number of licenses and bringing in revenue, increasing revenue, and then finally scaling out manufacturing. And those are all big inflection points for us.
That's excellent. Thanks for sharing that. Appreciate that. So what kind of specific technologies, actually, before I ask that question, I'm kind of more interested a little bit more about the hydrogel concept. I think it's important because there's a lot of impact, and I'm not sure how prevalent it is in the actual industry now. So in terms of like using hydrogels for delivering medicine, how common is that now for people or patients?
Yeah, so when you're looking at drug delivery, so let's think your small molecules. And when I say small molecules, I say things like acetaminophen, ibuprofen, dextromethorphan. These are all drugs that we buy on the shelf, right? And sometimes when you go to a grocery store, you hear about a drug, you're like, oh, there's a new 24-hour extended relief. I can go from, 2 pills a day to now 1 pill a day. I could just take, Claritin in the morning and I'm good all day long. The idea of drug delivery and formulating these kinds of drugs is not novel. It's been around a long time. In cells, it's something that is more, novel because it is such a nascent industry. The idea of encapsulating cells to protect them from the immune system, that's kind of been around, but no one's really thought about delivering cells because there really hasn't been a technology for that. And I think one of the interesting things we're finding in a lot of these conversations, and we're spending a lot of time educating folks on the fact that, look, you in drugs, we knew that when we delivered these, you gave these small molecules that they could work because they worked, whether it was on the bench or in an animal model. But then we had to formulate it to make sure that it hit the right target or hit the right functional window when you gave it to someone. And that's the same way with cells. We're seeing cancer treatments. I mean, there are approved cancer treatments out there for hematologic or blood cancers that are curing people of cancer. So you're like, okay, the cells can work. There are cures for cancer. But then when you look at all the other tumors that are out there, the other 80% or 90% of cancers, you say, well, those are really solid tumors. They're not blood cancers. We're still struggling to target and hit those cancers and basically get cells that we know are highly functional to work on those cancers. And so this brings kind of this new modality into kind of a tool. It's a new tool in people's toolbox to say, yeah, maybe I can now put this delivery package together with my cells, which I know should work, and be able to better deliver it, see the efficacy that I want, and hopefully see more successes in patients.
Yeah, I would just add to that we had to do a lot of educating because, and I kind of use the phrase that somehow the pharmaceutical industry, when it went to advanced therapies like cells, like antibodies, they forgot everything they knew when formulating small molecules. Like, you don't just take the acetaminophen, it's packaged in something. And yet they thought they could just inject these antibodies or the cells and they'd go where they needed to go and do what they needed to do. There was this old idea out there called homing, that the cells would know where to go. If you inject stem cells into the blood, they'll know where to go. Well, that's been debunked multiple times. So finally, the industry is starting to understand, no, we can't just hope that the cells will go where they need to go and stay there long enough to actually have an effect. And so we're seeing more interest now, but it's taken a lot of education on our part.
Well, thank you for educating, the pharma biotech community on this, and like a lot of your clients, especially. My only experience with hydrogel, or actually Matrigel, was when I was plating Matrigel and with intestinal stem cells. So we were growing intestinal organoids actually, and testing certain compounds with them in the lab.
Yeah.
And the hardest part was to ensure that they would continue to grow. So we were trying to play around with the media and different agents we would add so they can survive, and it was intense work. Every time your cells died, it was like a sad day. If they started growing and multiplying, it's a celebration.
Yep.
But yeah, so thanks for fine-tuning all of that for
and
delivery.
One of the things I think you might also appreciate, and I think any of the audience who are listening in, I mean, when you think about Matrigel, right, it's a very specific matrix that cells like to grow on, right? Your cells probably wouldn't be organoids if you just put them in a plastic dish, right? So that's kind of the same way when we think about our hydrogels. When you put them in the body, they like to be in these 3D-like environments, not necessarily as clusters, but cells talk to each other. They like to be together. The hydrogel kind of provides a backbone for them to rest on and to communicate and to grow. And that's kind of really what I think, bringing hydrogels from the dish, from the bench into the body, that's kind of what we're excited about. There's so much of that happening in the tissue engineering side, where you're building 3D organs or patches or kind of bigger technologies. But so many of these cell therapies, that's where we saw the opportunity to say, well, we want them to be smaller, more injectable, still leverage the fact that cells love to be in a 3D hydrogel, space. And we could take that further with some of our clients if we need to, where we could say we could actually modify these hydrogels to make them more amenable, provide a better backbone, provide a better environment for cells, make them more biologically active if that's what a client's program required. But that's not always necessary. Each cell is different and they all like a different environment. And so that's that, having the 50 plus hydrogel formulations gives us that customizability.
And we can also impregnate the hydrogel with things that the active ingredient needs, like growth factors for the cells. But also we can combine it with even small molecules. So think about a monoclonal antibody that's delivered to a target site with a chemotherapy along with it. I mean, we can mix things together with this technology.
Very interesting. Just a random thought I had: has there been experiments using hydrogels and matrigels specifically for like humans, like human therapies in space, like just to see how they grow? Or because I know gravity is a force upon the hydrogel when you create it or you plate them. So is there some sort of like idea that in space it might be a better place for cells to thrive in?
Yeah, we actually have an instrument in the lab that is zero gravity, right? So the cells are always moving in solution, never allowing to settle. Same kind of idea. And we use that a lot when we were in my research lab. We haven't used it so much here in an industry setting, but it does make a difference when they're always in suspension and not allowed to ever settle and basically be in a zero gravity situation.
Very interesting. So Lisa, it's been, a number of years you're running this company. How have you maintained the company culture throughout the years? You were around before COVID, through COVID, still around. So
yeah. So that is something that was very, very intentional. the company was Karthik and me, we sat down and wrote a handbook, an employee handbook, which seemed crazy at the time, but we were really passionate about, because in that handbook, we wanted to put together what is the culture. That was like our roadmap for the culture of Likarda. And, we have statements about culture in that, about how we respect each other. There's no bad mistake. We didn't want a culture coming from academia. You kind of see some labs sometimes where students are afraid to tell their mentor that the experiment screwed up or things aren't working. And we wanted just the opposite of that there's no bad idea. There's no bad experiment. There's no bad data. Even a failed experiment tells you something. And so we put that in place, and we've been really fortunate to be able to keep the same basic employees over multiple years. Now we've had, some whose spouses moved and had to move on, but our core set of folks are the same people we started with at the university. And for COVID, we didn't shut down. We, as a drug at that point, as a pharma company working on our own therapeutics in diabetes and osteoarthritis, we were allowed to maintain the labs open, but we made a schedule. So we knew when each person was in the lab one at a time, right? So your time was 3 to 5, somebody else's time was 8 to 11, so that there wasn't overlap of people. And of course, we took all the precautions. And then we allowed people to just work from home, because there's a lot of paperwork in this kind of industry, and you need to take the time away from the bench to finish all your paperwork. And another thing, kind of this goes back to where Karthik and I started, here in Kansas City, which is where we launched from. There's a very famous pharma company called Marion Labs. And it was started by a man named Ewing Kaufman. And there are many people here in Sanofi eventually acquired them, but there are many still old Marion Lab employees here. And when you talk to them, they only have good things to say about their experience at Marion Labs. And so I brought in the previous president of Marion Labs, Jim McGraw. He was a mentor of mine. And he also helped us understand, what did Mr. Kaufman do that was so unique and so powerful that he had such great loyalty by his employees. And it was a lot of, just respecting them, a lot of, things that you would normally think of. But he also thought about their lives and their family. And even back, and this was in the 80s, they had something called summer time. So every Friday at 2:30, they closed Marion Labs. Everybody went home from Memorial Day to Labor Day, so that everybody could get a jump start on their weekends with their families at home. And so we have incorporated that too. And we call it the Marion summer, Marion hours. But just little things like that we tried to take from Marion Labs and Mr. Kaufman's leadership and bring into Likarda.
It's a great example of learning from other companies and applying that to your own. That's really awesome.
Yeah, and I might just add, sure. we pride ourselves a lot on our internal culture, but obviously, we started out as a CRO, a service-based organization for our customers. Through our pivots, now we're supporting customers and clients. We actually really pride ourselves on being a great partner to our clients. I think one of the books that I read when we had first started Likarda thinking of a service organization was actually Delivering Happiness by Tony Hsieh, the founder of Zappos. And anyone can sell shoes, but he sold them better than anyone else. And it all came down to customer service. I think one of the things our team also gets really excited about is, internally we work great together, but we also really work to be partners with our clients. So when those projects come in-house, everyone gets to take kind of ownership of that. They get excited about working with the partner, get excited about updating the partners on the technology, and really fulfilling kind of the wants and needs that our partners are asking of us, even in those times of, failed experiments or high stress. So I wanted to just add that, yeah, our external culture also matters a lot to us.
I appreciate that, Karthik. And, just thinking about the challenges you might be facing as a company marketing this novel delivery platform, do you want to share a little bit about some of the learnings you've had over the years on educating clients, customers on this?
Yeah. So I think over the years, one of the things we found, when we were out there talking about our technology, we always were talking about it from our therapeutic platform that we're developing. And when customers started to recognize the need for cell delivery, then we had to quickly pivot to say, okay, we need to educate people on this. I think if we could start over, we would almost go back and say we need to be educating people from the start. We invented this platform out of our frustration as scientists for the lack of this kind of platform. We should have been singing that from the roof from the beginning, because I know, and we find now there's so many scientists out there who also had that frustration. And now to find our platform, they're like, oh man, this is great. I wish I would have known of this sooner. Now they have, when you think about the pharma sales cycle or the partnership cycle, you're looking at 6 months up to maybe 1 or 2 years. It's a long cycle. Drug development is not overnight. Some of these animal studies take 6 months. Human studies, to get the experience in humans, could take a couple of years.
Not to mention approvals and all that
yes,
with the FDA.
exactly.
Sure.
Yes.
Exactly. And so you only learn about these obstacles as you're going through them. And so, really in the last probably year or two, we've really tried to kind of push out more about education, trying to be out there, trying to get more info. And I think we're still building that educational package.
Excellent. Lisa, Karthik, I really, really appreciate your time today. And thanks for sharing all this with the audience and community and the VibeBio overall team. I think it's really important to get these stories out there, and good luck on your Series A round. How much are you raising again?
$15 million.
All right. Awesome. Anything else you'd like to share with the audience before we end here?
No, I think I really enjoyed this. Thanks so much, Ray. This is fun.
Awesome.
Yeah, I enjoyed the conversation. Thanks.